In this second part of learning and memory, I will talk about HAPS postulate again. Mainly, I will discuss the recent development in re-examining the HAPS postulate by looking at one aspect of a HAPS postulate, that is the timing of pre- and postsynaptic firing, so-called spike timing aspect of the plasticity. And we begin with this study from a retinal tactile system of Xenopus. This is a good system to study topographic mapping from one region of the brain to another region. And during the development, the retina ganglion cells sends axon to tecton to make connections. The projection of retinal ganglion cell is in an orderly fashion, that near neighboring ganglion cells are projected into near neighboring tactile cells in a topographically ordered manner. In the beginning, this projection is guided by molecular cues in the retina, complementary cues that interact with each other. For example, the FA receptors interact with afferent ligands in the tectum, and the gradient of this ligand in the tectum actually directs the axon to proper position in the tectum to make connections. However, this molecular cue in the beginning only set up a rough map, rough order between the ganglion cell and the tactile cells. As development proceeds, the sensory activity occurring within the system will refine this map so that there are more fine-grained point-to-point maps are developed in this system. Basically, the excessive connections are eliminated, and certain connections are stabilized. Now, the HAPS idea was used here very successfully to predict this process of refinement. We remember that the HAPS said that correlated pre- and postsynaptic activity caused strengthening and stabilization of the synapse, and uncorrelated activity eliminated the synapse. Cell fires together, wire together. We can use this idea, HAPS learning rule, to understand how that refinement of the retinal topic map occurs. For example, we know that natural images are spatially and temporally correlated, thus often simultaneously exciting neighboring retinal ganglion cells. A natural image means an object. The object always excites the near neighbor ganglion cell at the same time in space and in time. So spatial temporally correlated. So input from the same tactile cells are strengthened for nearby ganglion cells, but input from distant ganglion cells are weakened. So topographic map can be refined to more precise point-to-point map simply by HAPS learning rule. Showing here, the red axons innervating the same tactile cell. These cells are activated because near neighbor ganglion cells are activated by natural images. And distant, the green axons are eliminated because distant ganglion cells are often less correlated with nearby cells. So in the end, that the distant axons are eliminated, you get more precise point-to-point map between ganglion cells and tactile cells. Now, our questions 10, 12 years ago was the following. Now, HAPS idea is very useful to predict this transition, this developmental refinement. But when we talk about near neighboring cells firing together, that wire together, what timing are we talking about? Are they firing together within 10 milliseconds or within 100 milliseconds? When we say correlated firing, cell wire together by correlated firing, what correlation are we talking about? To determine precisely what is the correlation, the timing requirement. We perform direct measurements of the synaptic activity in the tadpole, in the Xenopus tadpole. We basically put an electrode into a single cells in the tectum that receive common input with these axonal input from different regions in the retina. So different retinal ganglion cells are making connections to this cell we're recording. Cell input 1, if we stimulate one of the one region of a group of ganglion cells here, they produce a synaptic currents or synaptic potential. Current is the opposite of potential. It drives us to potential. So here, synaptic current represents a synaptic strength coming off a group of synapses, group of ganglion cells, making connections to this cell we're recording. Now, input to a different region of retina also making input. So these two regions actually have converging input to the same site, same cell in the tectum. So we are recording from a tectum cells. We are exciting two regions of the ganglion cell that are making connections to the cell we are recording. So now our idea is, now if we fire these cells at different time, what is the correlation we require in order for their connection to be strengthened? What is the correlation what is the timing that we eliminate the connection? So here are the experiments. So we begin with a weak stimulation that produce two different input. Input 1, input 2. We stimulate weakly, they produce a depolarization, a synaptic EPSP, EPS2 EPSP. We produce correlated excitation, correlation within 10 milliseconds. So fire input 1, and 10 milliseconds later fire input 2. Now you do this pair firing repetitively for 100 times. And look at input 1 and look at input 2, their amplitude of EPSC, the synaptic strength. The synaptic strength remain unchanged at input 1 and 2 after repetitively correlated stimulation of these two inputs. Now note that this input did not fire action potential. They are subthreshold. So it's the subthreshold correlated input, correlated by 10 milliseconds, produce no effects. So repetitive firing presynaptic retinal ganglion cell from input 1, 2 cause no change in synaptic strength. Subthreshold, the conclusion is subthreshold excitation of postsynaptic tactile cell is not effective in modifying the synapse. Now if we increase the stimulation to input 1 and 2 by firing more ganglion cells so that the synaptic input can now reach a threshold, fire an action potential. So you produce action potential in both input. You look, after this repetitive pairing, you get strengthening of input 1 and input 2. Both are strengthened. Right? This is what HAP would predict. Presynaptic input, fire the postsynaptic cell, it contributes to the correlated firing, pre-post firing, and in both input, you have pre-post firing. Input 1 firing postsynaptic cell, input 2 fire here, and postsynaptic fire there. So correlated pre and post, you could produce synaptic potentiation, LTP. Persistent potentiation for a long time. This is 30 minutes, a long-term LTP. So this is the conclusion, that repetitive firing presynaptic retinal ganglion cell axon at both input causes synaptic strengthening at both inputs. Suprathreshold, that threshold that can fire action potential, this suprathreshold excitation of postsynaptic cell allows synaptic strengthening. You need a postsynaptic spiking to give you strengthening. Now, we can do more experiment. That's if we couple a weak input, one that only subthreshold input, with a strong postsynaptic firing of a second input. After this pairing, what happened? You see that input 1 and 2 are both strengthened. Now, the input 2, remember, this weak input by itself if we have only weak subthreshold input without spiking of the postsynaptic cell, we don't see any change. However, if you are coupled with a postsynaptic spiking, then this input will be potentiated. So this is the associative. Remember the associative LTP, that the weak input, by coupling with a strong input, you become strong. You get potentiated. So this is the conclusion. Then subthreshold subthreshold input 1 that comes before suprathreshold input 2. You get strengthening of both input. That postsynaptic spiking causes strengthening of preceding synaptic input. So this is important, that that this spiking can strengthen the preceding weak input. Now, the timing is important, comes from the last experiment, where the first input is strong, produces spiking. The second input is weak. The result is very striking. The result is that the input 1, the strong input is potentiated. This is exactly what you expect, right? Now, the correlated firing produces strengthening. The amazing thing is that the weak input now, even though they are associated with strong input with 10 milliseconds interval, this association did not allow them to be potentiated. In fact, they were depressed, right? Now, opposite to the case where they're in association by preceding the spike, they become potentiated. If they're in association with the spike but following the spike, they become depressed. Now, this potentiation and the depression are both NMDA receptor dependent. It's exactly what we expect for long-term potentiation, long-term depression. A process depends on NMDA receptor, because if you block the NMDA receptor with a drug called AP5, the LTP disappeared, the LTD also disappeared, right? So these are NMDA receptor dependent LTP and LTD. So now this finding, if our experiment was to test the timing, so we can now vary in the timing between input 1 and 2 and see exactly what the timing, how correlated they have to be in order for these effects of one input on the other. And what we found is that there's this striking window for LTP and LTD. That is, when the input comes in, a synaptic input comes in before the postsynaptic spiking, within a time window of 20 milliseconds, this is 20 milliseconds, you get LTP. This input will become potentiated after pairing. If the input comes following the spiking of postsynaptic cell, within a time window of 20 milliseconds, you get LTD. And this, both LTP and LTD require correlation, require the correlated firing of pre- and postsynaptic cell within 20 milliseconds. But the order of firing is important. In the case of LTP, presynaptic firing that creates this LTP, this EPSP, presynaptic firing occurs before postsynaptic spiking. You get LTP. If presynaptic firing, which generates this EPSP, occurs after postsynaptic spiking, you get LTD. So the same correlation is required. 10 millisecond correlation, 10, 20 millisecond correlation is required for synaptic modification. But pre before post, you get LTP. Post before pre, get LTD. And this is different from what original HAPS postulate was talking about. HAPS only concerned with correlation. Now, HAPS ideas of cell A firing cell B fits very well with this LTP, because cell A fire cell B means that the presynaptic input contribute to postsynaptic spiking. They have to be fired within a short period in order to contribute to postsynaptic spiking. So it gets potentiated, right? So this fits, half of this fits with HAPS idea, but the weakening part, it doesn't fit with HAPS' original correlation-based learning rule. So when we found this, in fact, that the LTP and LTD are generated depending on the timing of pre- and postsynaptic spiking. In fact, in the field of synaptic plasticity, several other investigators already reporting this. Henry Marcron and Bruce Sackman's lab, Dominic DeBan, Scott Thompson, Beat Gavina, and Curtis Bell, they all have reported in different systems that the LTP can be generated if the presynaptic stimulation occurs before postsynaptic excitation. And you get LTD if the order is opposite. Right? So we actually, when we found this, we immediately tested whether it's really postsynaptic spiking is involved. We directly inject currents into postsynaptic cell. Instead of using synaptic input to create this spiking, we inject current to create spiking and test the timing of the postsynaptic spiking in potentiate the synaptic input. We found that this circle, this timing indeed fits very well. The postsynaptic spiking is indeed generating LTP or LTD when they're depending on their order. So this led to a temporal specific rule in synaptic learning. This temporal specificity is very important for associative learning. I just talked to you about the Pavlovian conditioning in the last part. The Pavlovian conditioning in the dog, the meat is an unconditioned stimulus to induce salivation, but bell ringing is not a strong stimulus. It has no effect on salivation normally. But conditioning of a pairing of CS, the conditioned stimulus before US, before the meat is presented, you get salivation. But if you reverse the order, you present the meat before bell ringing. The dog doesn't learn because the ringing of bell follows the meat presentation. Associative learning has an order. The bell has to lead to meat rather than the other way around. Right? So inherent within associative learning, there's an order of association. And this is reflected at a synaptic level. And the synaptic learning rule now is an analogous timing requirement. Pre-synaptic firing before post-synaptic firing give you LTP. Opposite give you LTD. We now call this type of synaptic plasticity a spike time independent plasticity, STDP. So now, knowing the STDP, the spike time independent plasticity, we can now understand what is the rule for synaptic competition during development. The synapses that are innervating the target cell compete by willing in the competition by contributing spiking of the postsynaptic neuron. Those who contribute to spiking gets rewarded by potentiation. And there's a causality in this, that the presynaptic cell causes postsynaptic firing, then it's rewarded by potentiation. Also, the competition occurs within inputs arriving at the same time window of 40 milliseconds. Competition among a group of synapses, not all synapses, but only those synapses that are activated within 40 millisecond time window. That's the associativity. Now, like any competition in life, competition are always among nearly equals. Those inputs are arriving too early or too late within that are outside this window of 40 milliseconds are out of the competition. They are not affected by, their synaptic transitions are not affected. Only those arriving nearly equally at the same time within 40 milliseconds are competing with each other by firing the postsynaptic cell. If one fires relatively early, then gets potentiating. One fire relatively late, when the cell's already spiking, then one gets LTD, depression. So time is everything.